Hey guys, this week we will be covering Chapter 5, and we'll be going over thermodynamics, energy, and how enzymes help lower the amount of energy that we actually need in order for us to undergo our bodily processes. We're still laying the groundwork. This lecture, just like the chemistry lecture, is laying the groundwork to help us understand more about, or help us understand the processes of cellular respiration and photosynthesis that we'll be covering in the next 2 weeks. So, what is energy exactly? Energy is the capacity to do work, and without energy, we wouldn't be able to run, walk, or even breathe. Potential energy is energy that can be stored as fat and energy of position or configuration. The other kind of energy is kinetic energy, which is the energy of motion. These two kind of energies are constantly being transformed from one to the other. So here we have a diver that used kinetic energy to eat breakfast, and that breakfast provided potential energy that she could then use to climb the stairs. This kinetic energy turned into potential energy at the top of the diving board, and then as she dove, that potential energy of her position turned into the kinetic energy of her diving. So we get our potential energy from the food that we eat. And the energy in our food is measured in units called calories, which should sound pretty familiar, but it's not exactly what you think. So lowercase c calories is the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. Now, as you can imagine, this really isn't that amount or that much of energy. So instead, we measure the energy in our food by capital C calories, or kilocalories, and this equals 1,000 calories. In the United States, it will just say capital C calorie on our food packages, but if you actually go to other countries, they will say kilocalories on their food packages, and which is a little interesting fact. So, energy is regulated by the laws of thermodynamics, and there are two main laws of thermodynamics. The first law states that energy cannot be created or destroyed, but can change from one to the other, like we saw with the swimmer, changing potential energy into kinetic energy, and then back into potential energy. The second law of thermodynamics says that some energy will be lost as heat as it changes from one to the other. In between these conversions, energy is lost as heat. Another way that energy can be transferred from one to the other is when our bodies break down glycogen into glucose, and that glucose can then be used to help contract our muscles. So each time our muscles contract, energy is going to be lost as heat as the energy transforms from one to the other. This is pretty good as it helps keep up our body temperature, but this isn't exactly the most efficient use of energy since some of the energy from the food we eat is lost during this chemical transformation. The second law of thermodynamics also says that not only is some energy lost as heat, but also that the amount of entropy increases as well, as energy changes from one type of energy to the other. An example of this is within our house. As we live in our house, the amount of our houses get more messy, so therefore the amount of entropy increases as we expend energy doing whatever we're doing in our house. However, it's not exactly the most easiest place to live. In a messy house, we want our houses to be organized, and in order to decrease the amount of messiness in our house or decrease the amount of entropy, it takes energy to clean up our house or decrease the amount of energy within any closed system. As you can see here, a nice clean room takes energy to stay clean. It's so much more easier to throw our clothes on the floor, to not make our bed. So again, the natural tendency for any closed system like our room is to get messier, or to increase the amount of entropy, and to decrease the amount of entropy in any closed system, you have to expend energy. And again, life requires energy. Each action we take increases the amount of CO2 in the air, breaks down large biological molecules into smaller molecules as we use it. This greatly increases the amount of small molecules in our atmosphere, or another way of putting that, it greatly increases the amount of entropy in our atmosphere. So pretty much in biology, the more entropy we have, the more small molecules we have. In order to decrease the amount of entropy, we want to make those smaller molecules into larger molecules. And so that's where plants come into play. Plants are able to take those small molecules of CO2 and water and create them into a larger molecule called glucose, thereby decreasing the amount of entropy in our atmosphere. And just like our room, plants require energy in order to reconstruct those smaller molecules into glucose. That's why the sun is so important. The sun provides a constant source of energy for the plants to help create an organized mess out of all the small molecules in our atmosphere. Another reason that plants need such a large source of energy is because energy is lost as humans and animals eat plants, and transferring that energy from glucose into larger organisms. I don't know if you guys remember from our ecosystem lecture, there is going, in any ecosystem, there's going to be a lot more plants than there are animals. And that is because energy is lost as herbivores eat plants, omnivores or carnivores eat those herbivores, and then those top predators eat the herbivores or omnivores. And so therefore, we need that constant source of energy for plants to produce large amounts of glucose so that we can have those top predators in our ecosystem. We can see these patterns of energy and entropy at the cellular level. The natural state of any molecule is to go with its concentration gradient until it reaches an equilibrium on the inside of the cell and on the outside of the cell. This, even though it may not necessarily look like it, is increased entropy, is when we have an equilibrium of the ions on the inside and the outside of the cell. When all of the ions is on the inside of the cell or on the outside of the cell, this decreases the entropy of the cellular organism. So, therefore, it will take energy in order to move all of the ions to the inside of the cell or the outside of the cell. Another way of saying this is that it takes energy in order for ions to go against its concentration gradient, against the flow of the ions to the inside of the cell or the outside. There's a couple of different kinds of proteins that help the ions move into the cell or out of the cell. Channel proteins are the type of proteins that allow ions to flow down its concentration gradient. And because of this, channel proteins are not going to use energy. An example of a channel protein found within our cells are aquaporins, and these are specific for water molecules. So water molecules are going to flow into the cell and out of the cell at will. If the aquaporins are especially important within your kidney cells because it allows your kidney cells to regulate the amount of water that goes into your body or out of or to stay in your body or to be excreted out of your body, just so your body maintains the right amount of hydration. And then we have transport proteins. And transport proteins will move ions against their concentration gradient. And because of this, transport proteins will always be using energy. An example of a transport protein that's really important is the sodium-potassium pump that are found within our nerve cells. Without this particular transport protein, nerve conduction would be impossible, and we wouldn't be able to walk, breathe, or move around our house at all. So, transport proteins uses a form of energy called ATP. And ATP will be used by all cells for all of its cellular processes. ATP stands for adenosine triphosphate. The cell is able to use ATP by removing one of the phosphate groups, turning it into ADP, or adenosine diphosphate. The removal of this phosphate releases a lot of energy, since there's so much energy found within the chemical bond that keeps the phosphate group attached to that ADP. So the cell uses the energy from that broken bond to move the ions inside and outside of the cell. Because we're constantly breaking down ATP into ADP and phosphate groups, our body needs to constantly be generating more ATP, and it does this through cellular respiration. And so our body is our cells are constantly going through the cycle of breaking down ATP into ADP and phosphates, and then reattaching those phosphates to the ADP. So we talked about this a little bit before. There are different functional groups that are attached to molecules and proteins within our body, and the functional group will determine the chemical properties of that particular molecule. And ATP has 3 phosphate groups attached to it, and so that phosphate group determines the chemical property of ATP. So this here is an ATP molecule. We have the nitrogen-based adenine here, which should sound familiar since we just talked about DNA last week. And then we have this ribose sugar here attached to the adenine. The ribose sugar and the adenine together come together to form adenosine. Attached to our ribose sugar are our 3 phosphate groups, and the 3 phosphate groups together are just unstable. It makes ATP readily unstable. That is because there's just so much energy contained within those chemical bonds in between those 3 phosphate groups. And so the natural tendency for ATP is to get rid of that phosphate group so that it's at a lower energy state, which is really great for all of our cells because it can use that energy to undergo its cellular processes. And because the natural tendency of the phosphate group is to break away from ATP, it means that ADP releases energy really quickly since the cell needs it so much. When ATP breaks down into ADP and phosphate, it is called a chemical reaction. When the transport proteins in the cells regulate which ions go into the cell and out of the cell, this is called a biological reaction. So when this particular chemical reaction and biological reaction occur at the same time, it is known as a coupled reaction. And during coupled reactions, the energy released from the chemical reaction is given to the biological reaction that needs that energy. The breakdown of ATP and ADP causes other reactions as well, since it's able to give its energy from that chemical bond to a lot of reactants, which then allows the reactants to be formed into products. Another example of a biological reaction or a coupled reaction is when your muscles contract. Your muscles are able to contract because 2 different motor proteins, actin and myosin, use the energy from the breakdown of ATP to contract your muscles. So the arm-shaped myosin motor protein will use the energy from the ATP and then grab onto the actin filament, which is that thread-like yellow beaded string in the picture. It pulls onto that the actin filament, contracting your muscles. And then when you uncontract the muscles, the myosin then is detached from the actin filament, allowing your muscles to relax. So the constant breakdown of ATP increases the amount of entropy in our bodies. Our bodies need a constant source of energy in order to clean up this entropy, or in other words, make more ATP. The breakdown of glucose into ATP is called cellular respiration. And this is when one glucose molecule, which is C6H12O6, and then 6 hydrogen molecules are broken down into 6 water molecules, 6 carbon dioxide molecules, and our wanted product, ATP. Our constant need for glucose also needs to be supplied, which we do through eating. And we get all of the energy within our food from the glucose that plants produce through photosynthesis. And photosynthesis is just the complete opposite reaction of cellular respiration. Plants are going to take 6 water molecules, 6 carbon dioxide molecules to form 1 glucose molecule and 1 oxygen molecule, 6 oxygen molecules. Between photosynthesis and cellular respiration, this allows for there to be a constant flow of energy through nature that allows an ecosystem to be sustainable. Photosynthesis happens in the chloroplast organelle of a plant cell, and cellular respiration happens within the mitochondria of an animal cell. You guys can think of the chloroplast and the mitochondria as opposite organelles that work together to create usable energy for every organism. Humans are also involved in the cycling of molecules between plants and animals and in the flow of energy from the sun. We inhale oxygen, we eat plants and animals, and these energy-rich foods allows us to produce the ATP required to maintain our bodies and carry on activities. Because we are able to only consume a small amount of energy, we need different energy-saving mechanisms. And the main energy-saving mechanism our bodies use are enzymes. And enzymes are protein molecules that catalyze different chemical reactions in our body. There's going to be 1 enzyme per chemical reaction, so they're going to be highly specific. Last week we talked about protein synthesis. The enzymes are going to be made through protein synthesis, and there's going to be 1 gene that is used to create 1 type of enzyme. The molecule that undergoes the chemical reaction is called the substrate and will land in the enzyme's active site, which is again highly specific and so is shaped for that specific molecule. The reaction then happens in the active site when the enzyme changes shape. And when the enzyme unfolds, that substrate is now products and is released and is able to go wherever it needs to go. This then opens up the enzyme to receive more substrate and to create more products. So therefore, an enzyme can be used multiple times. So again, there is 1 enzyme per reaction, and so the active site on that enzyme is highly specific. You can think of it like a lock and key. This is called the induced fit model. Anytime the enzyme receives a substrate and undergoes a slight change in shape to accommodate the substrate, the chemical reaction then occurs, and then when the enzyme unfolds, it releases it releases the products into the cell to go wherever it needs to go. So just like with any biological or chemical reaction, enzyme activity needs to happen in some type of balance. So therefore, our body has developed natural processes to inhibit the enzyme activity if we have too much product building up in our cell. There is also unnatural processes that will inhibit enzyme activity in the cell. Poisons and antibiotics do this. For example, the poison cyanide will bind to and inhibit cytochrome C oxidase. And cytochrome C oxidase is involved in cellular respiration and makes ATP in the cell. So I'm sure you can see how this could be really bad for the cell. Antibiotics, like penicillin, will interfere with bacterial enzymes that will end up killing the bacteria in our cell, therefore limiting the infection in our body. There are a couple of different natural pathways within our body that will inhibit enzyme reactions. And these two different processes use feedback inhibition. The first pathway uses the abundance of the product to block the substrate from getting to the active site of the enzyme. And the second pathway is used when there are multiple enzymes within a pathway to get one needed product. The end product can be used to block the substrate from getting to the first enzyme. And it does this by connecting to another site on that first enzyme, thereby changing the shape of the active site and preventing that first substrate from getting to the active site and therefore shutting down the entire enzymatic process. So here we have our first process. We have our one enzyme, and it has created a lot of products, so the cell has decided it doesn't need that product anymore, and so it will shut down this enzymatic reaction. And it does this when the product will bind to another site found on the enzyme, thereby changing the shape of the active site, which does not allow the substrate to bind to it, thereby shutting down the enzymatic process, and products are no longer being produced. That product will disconnect from the enzyme when the cell decides it needs more of that product. In our second pathway, we have multiple enzymes being used. In this particular enzymatic process, we have 4 enzymes that are being used. So 1, 2, 3, and 4. Within this process, our first substrate will connect to our first enzyme, which will give us our first product, which will then connect to the second enzyme, giving us our second product, which will then connect to the third enzyme, giving us our third product, and then we'll then connect to our fourth enzyme, giving us our fourth and final product. And if our cell decides there's too much of this product, then just like in our first pathway, our final product will attach to another site found within our first enzyme, changing the shape of our active site on that first enzyme. Therefore, the substrate can't attach to the active site, and this shuts down the entire enzymatic process. When the cell decides it needs more of this particular product, this product will disconnect from that first enzyme, allowing the enzymatic processes to start up again. The chemical reactions in the body require a lot of energy. All of the small molecules in the body are disorganized, and so therefore there is a high level of entropy. We need energy to reorganize these molecules into the products our cell needs. The amount of energy needed to activate these reactions is called the energy of activation. And the energy of activation tends to be pretty huge. We would need to consume a lot of energy or a lot of food to cause these chemical reactions to occur. Enzymes are an energy-saving protein our bodies use, so we don't have to consume a lot of food. This is especially important in nature, where animals don't know when their next meals may come. So if you think of the energy activation as a really big hurdle, as shown by this red line here, you can see that it would take a lot of energy to start that chemical reaction to get the products that our cell needs. The enzyme lowers the energy of activation, or lowers the amount of energy that it takes to start that chemical reaction, therefore allowing those chemical reactions to occur a lot quicker and easier, so that our cells get the products it needs to really quickly. If our bodies didn't have these enzymes, then our bodies wouldn't be able to function at the level that they need to. So now we'll talk about how those two laws of thermodynamics can be seen at a cellular level. I'm sure you guys can remember from when we talked about cell anatomy, how there is a phospholipid bilayer around the outside of the cell. And this phospholipid bilayer is selectively permeable, which means it allows some molecules to go in and out freely. It prohibits some molecules from coming in and out. So therefore, some molecules require energy to be transported in and out of the cell. And there's 3 different kinds of transport that the cells use to get the molecules it needs. There's passive transport, which is when substances move from a higher concentration to a lower concentration and no additional energy is needed. There's active transport, which is when the substances move against its concentration gradient, and so energy is going to be required. And then we have bulk transport, which is generally going to be used by macromolecules, which are too big to use the membrane proteins to go in and out of the cell. And just like active transport, bulk transport also needs energy. Passive transport is also called simple diffusion since it doesn't require any energy. Molecules are able to move down their concentration gradient until equilibrium is reached. And again, because this is a natural tendency of any molecules, no energy is going to be needed. The type of molecules that generally use passive transport are the small nonpolar molecules like carbon or hydrogen, or like carbon dioxide or oxygen gas. So here so the reason our cell membrane is able to be selectively permeable is because of the polar head and the nonpolar tails of the phospholipids. The nonpolar property of our fatty acid tails prevents any polar molecules from going in and out of the cell at will. This allows the cell to decide what molecules move into the cell and which ones move out of the cell. Large and polar molecules require transport proteins in order to cross the cell membrane. Some transport proteins use facilitated diffusion, which is when the molecules are still able to flow down their concentration gradient, therefore these don't require any energy. An example of this, like we talked about a little earlier, those channel proteins called aquaporins, that is specific for water and allows water to freely move inside and outside of the cell. Osmosis is the specific term for the diffusion of water across the cell membrane. So for osmosis, you guys can think of the solute and water concentrations in terms of percentage instead of measurement units. So in this particular example, we have 100 milliliters of water in the beaker and then 100 milliliters of water inside our thistle tube. There is 10 milligrams of salt inside our thistle tube and 5 milligrams of salt found within our beaker. This gives us a 10% solute concentration in our thistle tube and a 5% concentration of solute or salt within our beaker. Another way of thinking about this is that we have 95% of water in our beaker and 90% of water within our thistle tube. With osmosis, water is going to flow down its concentration gradient from the beaker into the tube, thereby increasing the amount of water in the thistle tube, making it swell. The main reason for this is because there's too much solute or not enough water inside that thistle tube. And so when water flows across the membrane, it's going to lower the solute concentration and increase the water concentration. The actual amount of solute will not change, but the percentage of the water in the solute will. This natural tendency of water also greatly affects animal and plant cells. It is important that whatever environment the animal or plant cell is found in has the same solute concentration as the inside of that cell. When the solution has the same solute concentration as the inside of the cell, this is called an isotonic solution. And in this solution, the animal or plant cells will not lose water or gain water. And so therefore they will stay the same size. This is a really important fact to know for medical professionals who are giving you fluids to help maintain your hydration. This is also really important when you're donating plasma, as they give you a 0.9% saline solution to help rehydrate the person so that the red blood cells do not shrink or expand. And so your blood, your red blood cells are able to properly transport oxygen throughout your body. If the animal or plant cells were to be placed in a solution that has less solute in it, then it will have a tendency to swell because the water will rush from the outside of the cell to the inside of the cell. This type of solution is called a hypotonic solution. In a hypotonic solution, animal cells will lyse, and within plant cells, because the plant cells will still expand but won't lyse because of that cell wall. Again, that cell wall provides extra structure for the plant cell. Instead, it will create something called turgor pressure, which is again when that cell membrane presses against the cell wall. Anytime plant and animal cells are placed into a solution that has a higher solute concentration, they will end up shriveling because the water rushes from the inside of the cell to the outside of the cell. This particular solution is called a hypertonic solution. Hypertonic solutions will make red blood cells shrivel, which is also called crenation. And within plant cells, anytime they shrivel, they are said to have undergone plasmolysis and will have a tendency to wilt and cause the plant to die. Active transport is when energy is going to be used to move molecules against its concentration gradient inside the cell or outside the cell. Kidney cells use active transport to completely remove salt from the urine, as this is really important in helping maintain blood pressure. As you can imagine, this requires a lot of ATP, so kidney cells are going to have a lot of mitochondria near the plasma membrane, so it is able to transport that sodium. And then our final type of transport is bulk transport, and it's generally going to be used by macromolecules that are too big to use those transport proteins. These are instead going to be transported in vesicles, and this can be done either going into the cell or going outside of the cell. Exocytosis is when macromolecules are moved to the outside of the cell. So here we have our vesicle around our macromolecules. And our vesicle during exocytosis will bind to the cell membrane and releasing those macromolecules to the outside of the cell. During endocytosis, the complete opposite happens. Our cell membrane will form a vesicle around our macromolecules and transport it to the inside of the cell. There are some specific types of endocytosis. White blood cells will use phagocytosis to get rid of the bacteria in our body. And phagocytosis happens when a vesicle is formed around the bacteria, and then digestive enzymes are released into that vesicle to destroy the bacteria. The white blood cell will then give the bacteria to another type of blood cell that will help create antibodies against that bacteria, preventing future infections. Pinocytosis is when liquid is taken into the cell, and this type of endocytosis is less specific, and so many different molecules are able to enter into the cell at the same time through this process. Receptor-mediated endocytosis is a lot more specific than pinocytosis, which means it's going to require a receptor that is going to be able to accept a very specific molecule, which then will cause the membrane, which will then cause the cell to engulf that specific molecule to take it to the inside of the cell. Thanks for listening to this lecture, and as always, if you guys have any questions, don't hesitate to email me. Hope you guys have a nice week.